This is a short video describing synthetic biology and laying out how this is different to genetic engineering. So what is synthetic biology? According to the Royal Society, it is the design and construction of novel artificial biological pathways, organisms or devices, or the redesign of existing natural biological systems, basically tinkering with life to make it do what you want. You may be thinking that that doesn't sound particularly different from genetic engineering. Genetic engineering consisted mainly of replacing genes in an organism with those of another in order to get a desired function. Take the creation of golden rice, for example. Researchers wanted to create rice that produced vitamin A. To do this, they had to find the genes for the biosynthesis pathway in nature. The relevant genes were discovered in a daffodil and a bacteria. The genes were then laboriously extracted and purified from the daffodil, and also from the bacteria. These genes are then inserted into the rice genome. This creates a rice genome with the desired genes included and expressed as proteins. These genes then create the pathway for beta-carotene biosynthesis in the rice, which is a precursor to vitamin A. Given rice does not produce vitamin A, those who rely on it often suffered from deficiencies leading to malnutrition and death. The aim is to reduce rates of vitamin A deficiency by using golden rice, and it's free for subsistence farmers. This seems great in principle, however there are inherent problems that limit the possibilities and the efficiencies. Firstly, you're limited by what there is in nature and what you can actually get out. Getting the genes out of an organism is a laborious process. Secondly, despite the name, there was very little engineering. There was almost no standardization or abstraction, and this led to the processes being time-consuming and inefficient. For example, golden rice took decades. First, let's tackle problem one. How synthetic biology gets over the issue of being limited by nature and by what you can get out of an organism. Clues in the name, really. DNA synthesis has changed the game when it comes to tinkering with life and trying to repurpose organisms for our ends. So how does this game-changing gene synthesis actually work? You start simply with the 4 bases of DNA. These are chemically combined to create oligonucleotides of 6 to 100 base pairs. You can then combine oligonucleotides to create synthons of around about 1,000 base pairs. These synthons can then be assembled into devices, which can be many thousands of base pairs and consist of multiple genes. There are many clever mechanisms of gene synthesis out there. However, all of these start with the synthesis of oligonucleotides. The initial desired base is attached to a controlled pore glass by the 3' end, sits inside a small well. The 5' end of the base is protected by a tridal group, and the sides are chemically altered to prevent side reactions or forming double-stranded DNA. To get the process underway, the well is washed with acid to remove the 5' protective cap. The well is then washed with the next desired base, say thymine. The 2 bases bond, creating a 2-base single-stranded nucleotide. The well is then washed again with the next desired base, creating a 3-base single-stranded nucleotide. You get the idea. This is repeated multiple times until you end up with the desired oligonucleotide. The oligo is then washed with chemicals such as iodine and methylamine to remove the side protections and to add back the 5' protective cap. These oligos, there are lots of them in each well and lots of wells, are then pulled together to be assembled into synthons if that's what you require. There are multiple methods for synthon assembly. However, what we will discuss here is called Gibson assembly. This method, amazingly, can be carried out in a single Eppendorf tube, or in reality ordered online as it's faster and cheaper. For this method, oligos must be synthesized as DNA fragments with complementary ends, shown here by the blue and yellow colors. These oligos with their complementary ends are added to the tube or microplate and incubated with a Gibson assembly mix. In the mix, a 5' to 3' exonuclease gets to work on the DNA fragments. These chew up the 5' DNA, leaving 3' single-stranded DNA overhangs. As these overhangs are complementary, they therefore anneal, creating double-stranded DNA as desired. DNA polymerase then works to fill in the gaps, and DNA ligase seals the gaps. You're left with double-stranded DNA, which is a combination of the 2 DNA fragments that you wanted joined together. In a similar mechanism, these synthons can then be combined to create larger devices. For example, synthetic DNA can be inserted into plasmids, and this can be done by the synthesis company that you order it from online. A further step can be to combine synthons to create entire genomes. The Craig Venter Institute synthesized an entire 1 million base pair genome that functioned in the bacteria Mycoplasma. To do this, initially synthons or cassettes of 1,000 base pairs are combined in yeast cells. This creates 10,000 base pair assemblies, which are further combined. This creates 100,000 base pair assemblies, which again are further combined, creating the final genome. Given that it could take weeks to create oligonucleotides 20 years ago, this is an incredible feat. The time and cost to synthesize DNA has plummeted, and this is allowing us to take steps far beyond genetic engineering. So back to problem 2, the lack of engineering. One of the key aspects of engineering is abstraction, which basically means being able to work at a high level without everyone needing to understand the complex details below. Given the vast array of constantly interacting cellular machinery, this is important for realizing complex synthetic designs. At the most detailed level of the abstraction hierarchy, we have DNA. This is knowing and understanding what each and every section of DNA codes for. Next, we have parts. This is dividing sections of DNA into their functions, such as promoters, ribosome binding sites, the gene themselves, and their terminators. Each of these will have a different function based upon their DNA sequence, which is defined at a lower level of abstraction. Next, we have devices. This is a combination of parts that performs a specific function, for example, producing a color in response to sensing an environmental toxin. You do not have to understand the specific details of each part to create the device. You just need to understand what the function of each part is at a high level. Next, we have circuits, which are a combination of multiple devices. Here I'm going to give the example of a biological AND gate, where an output only occurs if you get two designated inputs. When both inputs, being factors that bind to promoters and induce transcription, are present, there is a protein produced from each device. Only if both proteins are expressed can they bind to create a new transcription factor. This factor then promotes transcription of a third device. Therefore, only if both the original inputs are present do you get the desired output from the third device. Using abstraction, complicated circuits can be designed by a person who does not need to understand every detail in the hierarchy. Finally, we have systems where multiple circuits are compiled to create multifaceted and complicated outputs that can respond to many factors and interact with one another. Evidently, with the volume of biological data out there, this abstraction is required to create complicated systems efficiently. But abstraction doesn't work without standardization. Standardization is key to modern engineering. To give an old example of this, back in the late 1800s, all screws were made by artisans to their own specifications. This meant there were hundreds of forms, none of which could be used interchangeably, creating massive inefficiencies. Only when Joseph Whitworth proposed standardized thread angles and diameters could these then be used by all. A modern example is building a computer. You can order parts for your computer from all over the world. You can then assemble these parts knowing that they will work in unison and fit. This is only possible due to standardization of specifications. Combining abstraction and standardization brings huge engineering efficiency. You don't have to know the details of how each part works. You just need to know how to fit them together and have the confidence that they will fit together. In biological terms, you don't need to know about the DNA, or even the parts, or devices, or even the circuits. You just need to know how to fit the systems to create the final package that you want. So what does all that we've discussed mean for synthetic biology, our modern genetic engineering? With abstraction and standardization, you can use the wealth of knowledge out there to combine systems to perform complicated functions. This computer-based visualization can then be realized through modern synthesis techniques. And when expressed in a cell, will hopefully give you the outcome that you were looking for. Hopefully, modern synthetic biology will allow us to produce new drugs and eco-friendly agricultural proteins and fuels from microorganisms. Far in the future, we may even be able to make nature do amazing things, like direct trees to create house-like structures. The possibilities are nearly endless.